It's just us?
For now, I don't know. We may have people coming in.
Cool, yeah. Thanks. How are you?
All right, how are you?
Good, good. Great, yeah. Okay, so maybe we should wait for other people, but I put plans together for the open source project with DevoWorm to see how we can move forward and how I could contribute. So, yeah. So, we'll wait for you.
There are you people. Are they there?
Hello.
Hi.
Hello.
Hello. Yeah, great. Welcome. So yeah, we didn't meet last week. We had I had some things as well as other people. So, there any updates from people?
I've been reading your paper, and since I don't know very much genetics, I'm surprised that I'm managing to read it. Fine.
Yeah.
I have some suggestions if you wanted to stay on the line till after or some other time.
Yeah, we could do that after the full meeting. Yeah, that'd be great. Yeah. Anyone else?
Okay, I'm going to go offline because I'm eating breakfast.
Okay. All right. Great. That's good. All right. So I guess Hussain wants to present on his things.
Sure. Yeah. Thanks so much. All right. Thanks so much. Yeah. As I mentioned previously, I wanted to get on board with. I'm still applying to jobs, receiving interviews, and talking to people in places about the best things to do moving forward across positions in research, tech, and software. They're very happy to see my contributions to open source, including DevoWorm. So this is a great opportunity for me to stay busy with something productive and relevant that's going to directly affect and help my career. So, right, getting right to things. Let me see if I can find my notes. Essentially, I'm looking to see if I can push the OpenWorm project forward. And I have a lot of data sources, but I'd like to create a project or do some work on data fusion and integration. I have a lot of data sources, but I'm struggling with finding out which ones might be the most relevant. I figured it'd be ideal to take advantage of CNNs, convolutional neural networks. They can be used for analyzing imaging data, for extracting spatial features, or recurrent neural networks for capturing temporal dependencies in the sequential data. I had a repository together just about some preliminary work that I'd done to get the project together. And I can see how things are. So I can show you guys how things are going.
Yeah.
And essentially, so I'm looking for any sort of feedback, particularly with what I can do to push things forward to really get things started, okay?
All right.
Basically, yeah.
So this isn't necessarily a fork of OpenWorm. It's kind of your own repository with things in it.
Yeah, my own repository. It's nothing forked, nothing like that, okay?
Yeah, that's fine. I mean, let me share my screen here, first of all.
Okay. My computer gets slow when I'm sharing something.
Yeah, so I'll just do it. So I have it up. Yeah, you can see it. So this has C302 in it, data, OpenWorm meta, some templates. So yeah, it has things in it. So what's supposed to be in the repository here?
Yeah, there's nothing. Okay, well, I've outlined in the README what I'm trying to do, pushing the project forward. Essentially, with this project, I'd like to, in many ways, be able to analyze and visualize the movements of the data and implement some sort of machine learning algorithm of some kind to optimize things or make things more efficient or effective, okay?
Okay.
So I figured transfer learning would be optimal for this project. And right now, there were some data sources that Bradley put me in touch with, but I'm looking for any sort of feedback right now on being able to push this forward or get things together, whether in the next week or the coming weeks, about just how I can contribute to DevoWorm in any way possible. So
yeah. So yeah, this has a movement analysis tool. So we have movement data that is in the. We have I don't know what the status of the movement data is right now exactly, but there's that. There's also data for embryo movement. So we have a lot of things that you can look at. It looks like this is you want to analyze and visualize movement. You want to use a set of machine learning algorithms, and you mentioned convolutional neural networks. So we do have the stuff in DevoWorm, which people have been working on for a couple of years now, and that includes things like I don't think we have a convolutional neural network in that collection of things, but we have RNNs and other types of things. But the choice of algorithm is it's kind of, you could use a lot of different algorithms if you wanted. It's not
right. So the best thing I think we focus on is what's a current objective or a current goal or problem that we might be needing to solve right now.
Yeah.
Okay, that's best.
Yeah. So I mean, there are a lot of things that we have that we talk about a lot. Yeah, I don't know what the best problem is to tackle. I mean, we'll have to think about that a little bit, because we could, we've used a lot of machine learning primarily to look at image segmentation, and then the GSOC project is creating graph embeddings from that. So like creating graphs.
Yeah, so that was someone else's GSOC project.
Yeah,
I remember, what was his name? Mongo? I forget his name.
Okay. Yeah.
Excuse me, I apologize.
Yeah. Well, there have been a couple of people who've worked on it.
Okay.
Yeah. But the thing we don't have there, of course, is movement. So there's a lot of movement in the embryos. I mean, we can do segmentation of static images. We've worked a little bit on movement, but it's kind of elusive. There's a lot of data on there are a lot of data sets where you could extract biological motion movements in embryos, movements in organisms.
Bradley, what's the time resolution of the embryo movement pictures?
Which ones? The ones that we usually use, or?
I'm not sure which. Any of them. In other words, how often do they take data? So that you could look at movements of individual cells over time.
Yeah, I think it's like millisecond resolution or something like that.
Oh, really?
Okay. I think so. Well, some of the newer data sets.
That should be.
Yeah. So there are a lot of cell tracking algorithms that we've also looked at, and there's if you go to the DevoLearn channel, there's a DevoLearn channel in addition to the DevoWorm channel on the Slack.
Yeah, I did look at that.
Yeah. And so there's this link to something called the Cell Tracking Challenge. So that's something that we've used in past years and the reason I bring this up is because it has data sets in it that are maybe useful here. And people, they want to track cells. Anyways, they have these data sets that they use to, track cells throughout development. So you're tracking their migration, you're tracking their position and things like that. That might be useful as well to have. The link is in the Slack, so it's there.
I'd be happy to contribute in any way possible. As I mentioned, my background's I have a strong background in data science, software engineering, even some experience in AR and VR if necessary too. We want to create a platform or tool to actually visualize this in 3D using spatial computing. That would be amazing too. So really, I think the best thing, especially asking you guys as scientists and academics, what particularly might you guys be looking for? What might be the biggest problems or objectives to really solve right now? And I can see how I can adapt myself to leverage things forward. I keep my schedule pretty flexible. I know how to, communicate needs and concerns as I need to with time and energy. So happy to get on board and volunteer. Yeah.
Well, can I suggest a problem?
No problem. Yeah, go ahead.
I was saying we still don't have a good handle on why a particular form develops, which obviously is a consequence of where the cells go and the orientation of cell divisions, and whether or not there's anything coordinated between cells. For instance, there might be some...
I'm just taking notes, right? Yeah.
Okay. So that's why I was asking about the time resolution. With millisecond resolution, you should be able to get that sort of thing. I don't think the cells move terribly fast.
Okay.
So you see, we've kind of broken the development into lineage trees, what we call differentiation trees. And in the case of the nematode, the two are equivalent for the most part. There are about 50 cells which form in pairs, So the differentiation tree is slightly different from the lineage tree. But that tree, in some sense, is wrapped in four-dimensional time.
Yeah, it is, yeah.
And we don't understand any of the rules for that.
Okay, so yeah, that'd be interesting to investigate. I can follow up and do a bit of research and background on this. And
okay, if you want, I can send you lots of papers on differentiation trees.
send as many papers as possible.
Okay, I'll need your email address.
Yeah, no problem. See, look, okay. Unfortunately, when I'm on these Jitsi and Google Meets, if I open up other programs, my computer gets really slow.
Oh,
I'm putting it in the chat. There's my email address.
okay, I'll put in my address. You can send me.
Thanks. I hope that's fine with you. It's just even if I open up Slack, believe it or not, my computer's going to get really slow.
So yeah, there, actually another set of problems we've had or tried to tackle are sort of visualizations, and especially spatial visualizations. So taking the data that we have and making nice visualizations of them. People have created like graphs and things like that are, not exactly what we'd like. So there's a visualization component that'd be really helpful, especially like creating a pipeline for, taking some idea, even not just data, and putting it into this four-dimensional space. So and of course you have the 3 dimensions, but then animating it over time and things like that.
Cool. Sounds great. All right. This is great. This is really, really great. Yeah.
Would you agree, Bradley? We really don't know how the differentiation tree is wrapped in four-dimensional space.
No, we don't. Yeah, the spatial part has always been tricky because I'm not really sure that the right data sets exist even now with a lot of the cell tracking people.
Even at that resolution.
Yeah. Well, knowing how to organize the data to do
for example, you could look at the orientation. Can you look at the orientation of individual cell divisions?
You can approximate it, but that's not...
You can approximate it.
Right.
Okay, so that would give you an approximate four-dimensional map.
Okay, thanks.
Okay.
So
are you aware, Hussain, that most of the cell divisions in nematodes are asymmetric? Do you know what that means?
So asymmetric meaning either the relationship from one side to the other does not reciprocate properly, or
no, you get one big cell and one small cell.
Yeah,
it's that simple in our naive understanding.
So like asymmetrically. Okay.
Okay, and you get about 50 cell divisions which are symmetric.
Yeah, the good thing doesn't.
What's the total number? 900 something.
Well, the good thing is that while it may seem confusing and complicated, the lack of degeneracy gives you a further way of separating signal from the noise. You can at the very least probe further to the data when you notice that, say, there's one big and one small one, and if they both, rather than had they both split to the same size of cells, then you might.
If you, for example, drew a vector between the nucleus of the small cell and the nucleus of the large cell, that would give you some directionality in space.
Yeah.
Okay.
It provides for
And I don't think we don't understand that directionality at all, as far as I know.
Okay. Oh, look, it looks like Susan is offering some papers as well. Perhaps whatever might be relevant, if anyone wants to contribute, I would really appreciate. So, if you wanted to, you could get a list of your papers, your resources, and send me an email. That would be nice. Okay. Well, thanks. That's all I wanted to share. Does anyone else have any other questions or inquiries? All right. Well, thanks, guys.
Okay.
Like I said, I'm balancing several different projects at once, but I can definitely devote at the very least 5 to 10 hours a week on an open work project. And I think that's plenty. So even working part-time open source. So I will just want to be clear. I am pretty frivolous in applying to jobs right now, and if I do receive a stable, secure position and it's confirmed, I may have to drop this. So this is meant to be. volunteering while getting some very valuable direct experience that I can leverage towards job opportunities. So that's where I am right now.
Well, you've achieved first, the STEM job offer. I've never heard of one.
Do you know how much effort they put into this? They literally had the URL domain, it was Trace Genomics, and they had a URL for Kaylee at recruitmenttracegenomics.com. They made their own domain just to scam me from a real company.
Yeah.
And Kaylee was a real person there, but they catfished her name of a different Kaylee. They were like, yeah.
So why would they do that?
Okay, here's the thing. There was one big thing they were doing, which isn't necessarily a red flag, but could have been questionable. They said they were going to send me a check to purchase equipment, like a laptop and a hard drive to use for the job. Okay? And I'm guessing what they wanted to do was to send me a fraudulent check, so I'd have to send some money back. And then when I go to cash it in to purchase the equipment, I'm stuck with a fake check. And they got some money out of me. That's a typical fraudulent check scheme. So whenever someone says, I'm going to send you a check through mail, you must be certain you can trust them. You must be certain that this is part of the job application process, okay? Otherwise, it's a scam.
We get checks in the mail for our grain all the time, and it's in thousands of dollars. Happens all the time.
I mean, I've gotten checks for freelance work. I've gotten checks for freelance work through the mail too. But they send me a legally binding contract, and I've spoken with them through video in person, so I know them as a real person. If they don't uphold their end of the bargain, or if the check is fraudulent, they can be legally held accountable.
Yes, that's true. Yep. Ours is under contract.
Yep. So here's the thing, Susan. There were two things. They send me questions through email interview, no phone call, no video, and they said they'd give me the job offer. I didn't meet anyone in person. Now that's not impossible. It is possible for people to extend jobs, through email alone. That is possible, but it's very concerning. The second thing was that they said they were going to send me a check and have me buy equipment myself. So I'm like, oh, you're not even going to meet me in person or call me, That's why the best thing to always do is to call with the company to confirm you are receiving a job before doing anything else.
Okay.
Yeah.
In case I want another job. Maybe not. Not after what you've explained. Forget this.
Yeah.
Yeah.
But you see what I'm saying? I showed their contract to many of my colleagues. They're like, yeah, it seems like a legitimate job. Congratulations. Nobody thought it could have been a scam. I had to call in to confirm. My friends who are professors, who are academics, they were like, congratulations, Trace Genomics is a wonderful place. It certainly is, but not this job offer. So always stay vigilant. Always stay cautious, no matter what.
When we get the grain checks, we always say, well, you take it to the bank and see if it bounces, then you know you've received it. So, yeah.
But even then, I don't know about the law specifically, but if you have a fake check and you try to cash it in, they could arrest you on that alone, even if it wasn't your intention. The best thing is to always be clear at first, Because even if you say to the bank or to the police, oh, but this wasn't my check, someone gave it to me. You don't want to be in that position. You always have to be careful first, okay?
Yeah. Yep. I
okay.
know.
Well, thanks, guys.
Yeah.
I do have to hop off right now. One of my other projects is asking for a phone call, but thank you so much for the information, and feel free to email me any other further communication or any other resources, papers, or anything else that can help me get on board with understanding the morphology and the morphogenesis of the cell division process. So that's a great place to start. Yeah. So thank you so much, guys. Bye.
You're welcome. Thank you.
Bye.
Yeah, so that's great. And then Susan mentioned that some of the work on biophysics and mechanics in development too. We haven't really touched on that as much in the papers that we've done. That may be something for future work. I don't know how well you can characterize that theoretically. I guess that's something that we could, develop some techniques around, especially in integrating it with the 4-dimensional tree.
Okay. Well, this was just how you could change morphology with cell shape change. And so it's, I guess, a part of this. And they're looking at a spherical epithelia, which, of course, our favorite salamanders have. So
Yeah.
that's why I found this yesterday.
Yeah. Great. Yeah. Thanks. So that's great. Thank you to Hussain for, pulling together a repository, where you can contribute some things. And that's really, what OpenWorm and even DevoWorm is about, is like people contributing to stuff. So it looks like he's actually, done some sort of documentation here with installing this this tool. It's not really all together yet, but there are different sources that are being put together. So and then this is how you would organize a repository. You put together all in the README. You put together the file structure, some things about details about contributing, and then of course the license. So that's description of kind of how that works. So I look forward to seeing what he can do on that. Okay. Any other comments or questions before we move on? Okay, so I'm going to get into some things today. We have a couple papers on C. elegans modeling I wanted to highlight. And they're like two papers that have come out recently. One brought to my attention by Morgan. And so this is MetaWorm. This is a project, I guess it's out of China. And they're doing something that is sort of similar to OpenWorm, but it takes a number of different approaches and puts them together. So we had a comment in the chat. Oh, okay. Pakhi, would you like to ask your questions now? You can, if you want to, you can do it. Okay, you can type it in the chat if you'd like to. So that's fine, and then I can come back to it later. That would work. I usually read them out of the chat and then answer them. So that please feel free to do that. So this is this MetaWorm paper. So this is again some Chinese group. I'm not familiar with their work overall. Focused on artificial intelligence and biomedical imaging. This is MetaWorm, an integrative data-driven model simulating C. elegans brain, body, and environment interactions. So they're interested in this movement, or looking at movement, I guess. We'll see what they have to say in the abstract. So the behavior of an organism is profoundly influenced by the complex interplay between its brain, body, and environment. So that suggests that they're looking at how the worm interacts with its environment. And in C. elegans, that means you're interested in how the worm is moving. And so existing data-driven models focusing on either the brain or the body environment separately, which I guess means that it's focused either on the connectome or on the body simulation. So in OpenWorm, for example, we have C302, which is focused on the connectome, and we have cybernetic, which is focused on the biophysics of the body. So there's two different platforms there. They're not well integrated in the technical sense, and so this is kind of, I think, what they're talking about, that those don't exist as a unified thing. A model that integrates these two components is yet to be developed. Here we present MetaWorm, an integrative data-driven model of a widely studied organism, and that's C. elegans. This model consists of two submodels. So there are two compartments to the model, the brain model and the body and environment model. So, we have in OpenWorm two separate programs that do this, C302 with the connectome and cybernetic with the biophysics simulations. What they're doing is creating a single model or a single program that has two compartments that simulate these things. So as an aside here, you can have models of different compartments, and the idea is that the compartments simulate things sort of as a separately, as a module, and then it gets put together as in the same program as a single output. So you might, for example, simulate the brain or simulate the connectome, have a simulation running for that, and then have that connected to a simulation for biophysics. You're not, you don't have a single sort of model that takes an input into the body and then goes into the connectome and then comes out. You might do like a physics simulation in one compartment, like an interactive or a network simulation in another compartment. And then those two compartments would communicate with each other, exchange data, and then the output would be some summary of one compartment and a summary of the other compartment. So, there are ways to do this where you can get a concurrent answer. If you have two models or two programs, it's not easy to do that. You were going to ask a question, Susan?
Sorry, no.
Okay.
It's okay.
Okay, yeah. So the brain model was built by multi-compartment models with realistic morphology, connectome, and neural population dynamics. So they actually, even within their brain model, which is the connectome, they had several compartments within that. You have a connectome and neural population dynamics. So each neuron, you have the connectome, which is a network of neurons. Those neurons then themselves are not neurons, they're just nodes in a network. But those nodes then have like population dynamics. So they have some sort of simulated electrophysiological activity, and you can actually take those as a whole, as they do in neuroscience of population dynamics, and look at the mean output of that. So this was all based on experimental data, so the model was validated with experimental data. Simultaneously, the body and environment model employed a lifelike body and a 3D physical environment. So this is where you have something like cybernetic, where you have the worm embedded in a medium that you're simulating. So it could be like a gel, or it could be like a liquid, and you're simulating the movement within that medium, the resistance against the body, and the movement of the muscles and things like that. It's a very different simulation than what we have for the connectome. And so this allows you to quantify the behavior. Through the closed-loop interaction between the two submodels, MetaWorm faithfully reproduces the realistic zigzag movement towards attractors observed in C. elegans. So this is one of the things you find in C. elegans behavior, or you find these stereotypical movements. Some of them are what they call zigzag movements, which are where the worm undulates back and forth. There are other movements that you can produce, or that the worm produces, that we can simulate, but this is probably the easiest one. So it produces this one movement. Notably, MetaWorm is the first model to achieve seamless integration of detailed brain, body, and environment simulations, enabling unprecedented insights into the intricate relationships between neural structures, neural activities, and behaviors. Leveraging this model, we investigated the impact of neural system structure on both neural activities and behaviors. And so this is just, it helps us understand the behavior of the worm and some of the things going on that generate those behaviors. So this is, they kind of go over some of the details of these types of models. They kind of cite some of the types of data-driven models that exist to simulate either the brain or neuromechanical models for the whole organism. So they give examples of the mouse primary visual cortex, which is citation 1, the mouse striatum, which is citation 2, rat somatosensory cortex, which is number 3, and then for the neuromechanical models, which are these kind of models where you're merging the connectome with the body dynamics, we have Drosophila and rodent, 4 and 5. And then, I like this, OpenWorm Project 6 aimed to simulate past tense, C. elegans years ago, despite its detailed brain model and body model, work separately. So apparently C. elegans, or apparently OpenWorm, is not doing anything anymore. It's not true, but that's like they kind of say there's a need for an integrated model. So no data-driven model exists that successfully integrates these elements. So this is one of the things that they. So let's take a look at those citations to see if we can find.
Where is this in archive?
This is this is bioRxiv.
Okay, you can send them a correction to that sentence for the final version.
Okay, yeah. Let me see if I can go down to the bottom here to find the citations. I wanted to show kind of what citations are in the literature for this. Okay, so of course, one is Hodgkin and Huxley. This is the original paper on biophysical neurons. So, or one of the original papers on simulating biophysical neurons, Hodgkin and Huxley. And so they lay out the Hodgkin-Huxley model here, which is a model of, we have a tutorial of it in OpenWorm that you can do. And it just demonstrates kind of how action potentials work and how ion channel dynamics work. The second paper is Nicoletti, which is biophysical modeling of C. elegans neurons. That's from 2019. This is the single ion currents and whole cell dynamics in AWCON and RMD. And those are specific neurons in the C. elegans connectome. So people are doing like their single cell recordings. They're doing, we have these models, these classic models that we can apply. Grove and Sternberg, which is this, the Grove and Sternberg, they're from a group at Caltech where they do a lot of simulations. On Saturday, we were talking about some of the stuff they've done in Drosophila, and they've also done things with C. elegans. So that's they're referring to there. 4 is of course OpenWorm, the C302 paper from the Royal Society issue that was put out in 2018, and then there some other papers on connectomes and some other things. So we have a lot of resource. There aren't that many resources on these sort of whole organism simulations, but what would be helpful here, I think, also would be to have a nice review of that. I'm not sure if people have actually done this, where they look at the different ways in which different groups have simulated this. I know there are a number of other papers which you could cite that would give insights into this as well. When Morgan actually mentioned that they were using some sort of model that he was interested in. I don't know if Morgan's available to.
They're using finite element modeling.
Okay. So finite element modeling for the body.
Yeah, it's just it's a physics model. Not that they have to in the. I was curious why they were actually using it, or like how much they were bringing in, kind of soft body physics, in it all.
Yeah. Did you get the impression that they didn't specify why they were using a finite element model, or was it that they?
It would be natural. I didn't actually. Sorry, I'm getting ready for school run here. What departments are they in? Like if they're coming, if they're coming from mechanical engineering, then it would just be. It would probably be the tool that they know. You know what I mean?
Yeah.
Like as opposed to a kind of conscious decision.
Yeah.
But as you know, there are plenty of groups that do all sorts of robotic movements without finite element modeling.
Right.
But I think it opens up, or again, if everything is there and available,
it opens up some other things that potentially we could do.
Yeah.
Sorry, I'm not in front of my computer.
Yeah, that's okay. Yeah, that's good. The reason I ask about the finite element models is because we've talked about, very early on in the history of the group, we talked about finite element modeling and how that might be useful in some of the developmental stuff. So this was, back in the 2010s. So, I mean, I don't know what, the outcome was, I guess, negative in the sense that we didn't adopt that method. And it does take a little bit of expertise to use a finite element model. I know Susan probably knows something about that, but but there are a lot of options for doing modeling of this type.
And I'm using COMSOL, and it's being a huge pain. It's finite element analysis. Hussain, have you used COMSOL at all?
You're coming in and out. I was on a phone call with the job opportunity.
Oh.
But no, I've not used. I've used consoles like virtual consoles before. Is that what you mean? Or you mean the console log?
No, she's talking about a particular finite element package.
No, I haven't used that.
No, okay. Well, it doesn't like to do Slack tables, and unfortunately, that's what cells are all about. So it's very annoying. I'm going to try to correct it this week, but it's just not ideal. And also, if you have something under compression, it won't do something in tension, and your rod falls apart. Well, that's fine. It's a microtubule, and it falls apart, and it rebuilds itself, and that's great. But the COMSOL package just says it's an error in your building craft. It's not a building, it's a cell. So yeah, I'm having some difficulty with it because it's too restricted.
Yeah.
I mean, I'm happy to look into that too. If you want to email me your concerns too.
Yeah.
Yeah, well, I'm going to. Yeah, I should list them because I need to hand something in.
No problem.
And so yeah, I could send you the list of problems with it.
Sure, my email is in the chat, so go ahead.
Okay, all right, thanks. Yeah, I'll look for it here. Yeah, there we go.
It's the S Hussain Ather, that one.
Yeah.
So yeah, that brings up also that there are actually other packages that explicitly model soft body dynamics. So
if you know of anything that explicitly models tensegrities, let me know. The one that was available online is not really available. It seems to be an older package using Java, and it doesn't want to work.
Yeah.
I can't download it. I can't use it online. I can't download it. I was going to use MATLAB, and apparently there's something called Grasshopper that actually people have used to make tensegrity models, but it looks like Symlink in MATLAB. But anyway,
yeah,
that's about all I know.
Yeah. Okay. No. Yeah. It's one thing I've learned recently, especially since I started dipping into like more general legitimate software engineering, is how much science and research is really held back by these sorts of things. That we don't have enough good software engineers who know how to build software. I know, especially when I worked in bioinformatics, like it was such a like a I don't know, I don't want to say a disaster of a discipline, but just so many things that did not depend on the people with PhDs in biology, but people with software engineering experience to figure out. So
Yeah, well, that's what this is.
Yep.
It's causing me a lot of problems, including making my PhD into a master's, apparently, because I'm not making enough progress. Don't have enough time. I said, but it's multidisciplined. Doesn't matter, is it multidisciplined, and there's 5 disciplines involved.
Yeah. I'm sorry, Susan. I know I said I would get back to you about some of this stuff too.
Yeah. Oh, it's
please.
fine. I know where its faults
Okay.
are right about now. But
Sure. But it'd be really interesting because, bringing this down to cellular level and thinking about transcription is would be of real interest. So I can also try some of the Slack forums for finite element packages that other open source finite element packages too.
yeah, well, if you can
If
find
you've
one
got a
that does this Slack cable thing.
if you've got a bit of a description that you can post somewhere or send to me, then I'm happy to put this in various Slack forums.
Okay. I will. Yeah, I'll just. How about if I send everybody my complaints?
Yes.
List of grievances, yeah.
Yeah.
Yeah. So that's good. And I think you're right. There are a lot of packages that you could do like soft body modeling, and I think this holds true for OpenWorm as well. It's hard to use a lot of the packages, outside of a small range of sort of use cases, and so you have to build tools. Sometimes you have to build new tools to get exactly what you want, and that's not great. We need more. We need more.
Yeah. I mean, it came up with that virtual Drosophila that you talked about Saturday.
Right.
Like they added things to Mujoko.
Right.
That were very physicsy, just to handle the kind of the dynamics that were going to be with this particular kind of flying motion.
Right. Yeah, it's but the trick is being able to add things. Yeah.
Yeah, I mean, well, I think it shows you that you needed a really big team because you looked at that list and there was people all over, and I think it was very much like who's got this very specialized expertise.
Right. I think we should have that conversation at some point, though. I think especially Hussain could be helpful here, and kind of maybe just even enumerating the things that are needed in like this sort of community tool building or these open source tools for doing these kind of simulations.
For sure.
It's definitely a barrier to have tools that you can't use where you want to use them, and then you have to build a new tool. Yeah.
Well, I was also wondering how useful, basically their work would be in terms of sharing overlap with OpenWorm.
Yeah, I think there's some like new stuff that they've done. Of course, like they've done a lot of stuff with modeling, with like different multi-compartment modeling and things like that. And so, I didn't read through the whole paper, but it looks like they've done some really good work here, especially in terms of modeling a body that can interact with the environment and the nervous system. So, they always try to pick the best tool for the job. Sometimes you have to create your own tools to do that. So like if you implement, say, a finite element model, you might have to do that. You can do that maybe out of a package. You have to homebrew the whole thing and start from scratch and then apply it directly to your problem. So, but it looks like they did the soft body simulation and finite element. I think the soft body simulation, we've been kind of encountering a lot of cases where, and of course the embryo is a soft body. It's not a hard body. And so to really capture the dynamics properly, you do need to have this soft body aspect to it. And whatever that means. I guess that means that you can model the cells not as square things, but as squishy things or irregularly shaped things. And that's one of the things, and I don't know if they're actually doing that here, but that would be kind of, adding the physics in like that is really what's needed. And so, there's good stuff in here. They have target body reference coordinate system. So they have a lot of stuff in here. It's really interesting. Okay, so that's good. And then they have some supplementary movies. So if you go to, there's actually bioRxiv preprint. I'll open up the link here. And this is the bioRxiv stub, so they have, I don't know where they have the supplemental material. Here it is. So this is, of course, you can get access to the supplementary material. And I have movies here. I'm not going to open up the movies because it's going to be, I don't know how it's going to respond, but basically we have movies here. There's simulations, and you can check those out. Yeah.
Yeah, that'd be. I'd love to talk about that. I haven't actually checked out if they've got a GitHub repo or like which particular packages they depend on. I think it would be great to look at the same time at what Josh Bongard uses in for his course in terms of tools.
Right.
Because as someone who's been so focused on evolutionary robotics for so long at a software level, I think his choices would be really useful and enlightening.
Yeah, so there's an event coming up this week, Embodied Cognition, and Hussain's presenting at that, and so am I. So, but Josh Bongard, I think, is presenting there. And there are a lot of people interested in soft robotics there.
Oh, that's cool.
Yeah, so there'll be a lot of new stuff maybe from people. We'll see what the state of the art is on that. So to talk about the other paper here for today, this is from Scientific Reports. This is learning the dynamics of realistic models of C. elegans nervous system with recurrent neural networks. So what they're interested in is kind of a model of the connectome, which we've talked about, and using recurrent neural networks, RNNs, to model this sort of thing. So this is a group here, I think they're Portuguese, and they, so let's read the abstract. So given the inherent complexity of the human nervous system, insight into the dynamics of brain activity can be gained from studying smaller and simpler organisms, which is the C. elegans justification here. So, while some of the potential target organisms are simple enough that their behavioral and structural biology might be well known and understood, others might still lead to computationally intractable models that require extensive resources to simulate. So this is one of the things about simulation as well, is that you have to make the problem tractable. So you can't just simulate anything you want. You have to pick up a problem domain, or ideally, you have to set up the problem so that you can actually run the thing, and it gives you insightful results. Since such organisms are frequently only acting as proxies to further our understanding, often one is not interested in the detailed evolution of every single neuron in the system. Instead, it is sufficient to observe the subset of neurons that capture the effect that the profound nonlinearities of the neuronal subsystem have in response to different stimuli. So that's kind of what their justification for how they're approaching this. So here they're considering the C. elegans connectome and the C. elegans nervous system. They want to investigate the possibility of generating lower complexity models. So they're not interested in the whole, everything involved in sensing something, processing it in the connectome, and then giving a behavioral output. They're interested in isolating certain parts of the nervous system so they can build these lower complexity models. They capture the system's dynamics with low error, using only measured or simulated input-output information. So this is where you really have a very sparse model. And this is, of course, because sometimes, the models that we just saw are going to take a lot of computational resources, having all these compartments to simulate. So being able to do a very minimal model is always good, just to be able to capture some of the essence of the system you're interested in. Such models are often termed black box models. So black box models mean that you don't specify the details, you might simulate neurons in a system. You don't specify the biophysics. The biophysics are implied, which is the case in most neural networks, we can say, okay, this looks kind of like a neuron, but it's not really, but it's good enough. So that's why, we use black box models. And of course, there's a whole argument to be had about whether that's sufficient, but that's beyond what they're doing here. We show how the nervous system of C. elegans can be modeled and simulated with data-driven models using different neural network architectures. So now they're using data inputs from, I guess they could simulate them or get them from data sets, and they have different neural network architectures they're using. Specifically, we target the use of state-of-the-art recurrent neural network architectures, such as long short-term memory, which is LSTM, and gated recurrent units, and compare these architectures in terms of their properties and their accuracy. And they use root mean square error as a way to measure the error in the model. So there are these different architectures of recursive neural network. They test these different architectures and they look at the error and how it minimizes the error, as well as the complexity of the resulting models. We show that gated recurrent unit models with a hidden layer of size 4, so this is where you have like the number of layers and the architecture that are specified, and they tested a range of those, and they were saying that the gated recurrent unit model of hidden layer size 4 is able to accurately reproduce the system response to very different stimuli. So this is where you have different types of stimulus coming in, which model basically replicates the C. elegans connectome the best, which one fits what you might see in the biology the most faithfully. We furthermore explore the relative importance of their inputs, as well as their scalability for more scenarios. So this paper, I wanted to bring this up because it kind of highlights this idea of minimal modeling, but also, how you can use neural networks to kind of get at that and how to simulate the connectome. So they actually used the neuron simulator for this to build this connectome. They obtained a collection of synthetic data sets to represent the system's response to different input signals. So you want to have different types of stimulus to see if you're getting something that the connectome might be able to handle. So you don't want to just try it on homogeneous training data. So this is so they go through their work here, and so this is sort of their modeling methodology. They have the connectome, they have the ML framework, they have a model, they have predicted outputs and true outputs. So they can simulate the connectome, get an set of outputs, then they can use their models using the same input data, and they can build predicted outputs. And then the idea is that you want to minimize the error of the predicted outputs with respect to the true outputs. So that's what they're doing here. And so I think that's a good lesson in how one can do that kind of a study where you're looking at, say, biological information processing versus something like a simulation, and kind of comparing the two and say this is realistic. In the first paper, of course, their goal also was to be hyper realistic and to get kind of lifelike results. So, and they use similar methods. They use a finite element model, but they're also trying to minimize the error with respect to the biological system. All right.
Minimizing the error with respect to the biological system. Like, did they compare the movement they're getting to the movement in the biological system, or how do they do that?
Yeah, I guess you quantify the output. Well, in this case, in the second paper, they're simulating the connectome. So it's actually not it's biological in the sense that the simulation is on a connectome, which is biological in its origin. So that's actually kind of simulated, semi-simulated. They're getting a result for that. And then they're taking their model, which is the neural network, which is not biological, although it does process things. The whole principle of neural networks is that it works kind of like a brain. I mean, there are debates about whether that's, what the similarities and differences are. but they basically can produce an output, and then that output gets minimized over time as you train. You train the model, it gives outputs. You get, over time, it converges to some solution, and that solution needs to be close to the observed values for, say, like the connectome simulation. So that's what their rationale is there. In the first paper, we can actually get data on movement, and we can quantify that with, tracking of the worm and things like that. And we can get, as long as we can create a curve, we can say, this is what the biology is doing and producing as an output. And then we can simulate that and actually see if we can match that.
Yeah, okay, yeah.
There's a fair amount of systems biology operating on that principle, that if you simulate, if you can simulate a system, you want to measure, it's a measure of the real system, and then estimate, kind of get close to that performance.
Yeah, well, I'm doing that with stress strain curves. Or I'm supposed to. Like find a real system and then recreate the curve somehow.
Right.
I have some ideas, but whether they'll like it or not, I don't know.
Yeah.
I showed them a 2D model last week, and they were going, "What's this?" I said, "I asked if I could go 2D. You said yes." So anyway, yeah.
Well, yeah, I think for figuring out the methodology, 2D is probably better because it's less, you don't have to invest a lot in getting you want to see what works in 2D before you can,
Before do the 3D, yeah.
Right.
But I've already done 3D, and of course it sort of fell on its face, partly because of the
3D gets really
streams.
Yeah.
Pardon?
3D gets very intensive, so.
Yeah, I don't get the behavior, the tensegrity behavior out of a 2D model as readily as I do out of a 3D model. But I did manage to get some tensegrity behavior out of a 2D model, so that was quite good, especially since I was using springs that are linear. And anyways, they're saying, that springs are linear, right? And I'm going, yeah, they're linear springs. I'm just trying this 2D model. See, I got a J curve. Anyway, yeah. But the trouble is, I was pushing a rope, apparently.
Oh, okay.
You don't do that. The mechanical engineer on the committee was going, "What? You need to take more mechanics." No, I apologize for pushing a rope, okay? Yeah. Anyway.
Yeah. Okay, so that's great. So that's the recent C. elegans modeling papers. I think that's useful for a number of things. And I wanted to mention also that I'm putting together a 10th anniversary slideshow on DevoWorm. So next month is DevoWorm's 10th anniversary. So we've been doing this collaboration for 10 years. And I wanted to put together like a summary of everything. And it's kind of interesting going back and looking at what we've been doing in the past and present. And it's, yeah. So hopefully, I'll try to produce, I think probably one of the meetings in April will talk about it more. And, maybe that'll bring some old topics back up to the top. So
okay, well, next week I might not make it because I'm going to Saskatchewan.
Okay, yeah.
But yeah.
So I think Pakhi wanted to ask some questions. I don't know if you are able to talk or if you want to put them in the chat.
Here, I'll turn this off.
Okay.
It doesn't help. There.
Hello.
Yes. Hi.
Yeah. Hi. Hello, everyone. I hope I'm audible.
Yes.
Yeah. So I had a couple of questions. I went through the DevoGraph repository and I'll start with the code first. So I wanted to ask, like under the stage 1 folder, there is this RCNN mass segmentation. So there are a couple of notebooks within that folder. So do they need to be like converted to complete pipelines to be merged with like the DevoGraph folder, like the models over there? Like, is that like a requirement?
It's not a requirement, but may have. The thing is that some of these things we've been working on them in pieces. So stage 1 is basically cell segmentation. So we can do that. There are a number of different tools, and I've been trying to clean it up, but it's been tough because we've had multiple people kind of contributing. I don't think for the sort of the proposal you need to do that, but it would be something if you wanted to propose it as part of the project this summer. That would be good. Because what I want to do this summer is I think we have things for each stage at this point. And to try to get those into a pipeline or try to optimize those in some way. That's really kind of what we'd like to do. So if you need to do that to get it running, okay, but that's, you don't have to do it right away. I'm not really sure what the state of it is though.
All right. Because like the third point mentioned in the problem statement was like tightening the DevoGraph with the DevoWorm. So I wanted to ask if we have to integrate it in the pipeline. Okay. So other thing I had was like we are basically monitoring the embryonic development of C. elegans, right? Correct me if I'm wrong.
Yeah. So what we, at least at this point, what we would like to do is have a 3-stage model where we take stage 1 would be like cell segmentation. So we have some data sets that we can use for C. elegans embryo, and we want to segment those cells, and then we want to create like a set of cells, and then we want to plug that in to stage 2, which is creating graph embedding. So once you have the cell segmented, you have features that you can then use in the graph embedding. Then the graph embedding is something that you can produce in, I think we have some software for that in there, but there are other, basically that's forked from some other tools that exist out there. And there are a lot of tools that can be integrated. So, there's a lot of room for improvement on stage 2. Stage 1 really is, there are tools there, but we have done a lot of work on the DevoLearn platform. So those are available. Stage 2, there's stuff available from the outside world, from the open source world. And that's creating the graph embeddings. And then stage 3 is like creating visualizations. Himanshu last summer, Himanshu Shobal worked on topological data analysis as a way to visualize the graph embeddings and the results of that. So, what the project basically is taking those 3 stages and improving different parts of them. Stage 1 certainly could be improved, but stage 2 and 3 are probably the least developed and probably need the most work. So any kind of combination of things that you propose to do on any of those things is acceptable. I would like to see if you, before you submit your proposal, that you give me sort of an outline of what you want to do. I can, in very specific terms, so I can get a sense of what people want to do, and if we need anything before we start the coding period to get up to speed. Because some of these things, like I said, some of these things may be very easy to do. Some of these things may be very hard to do. And so it's important to be kind of gauge how much work something is going to take, because that's important, because you have, a finite amount of time to do it in. So.
Yeah, I understand. Actually, I came across a paper like over a month ago for some other project, and I wanted to share it. Maybe I can post it in the chat.
Yeah.
And basically, in that paper, they are like they have proposed like developing a neural network that basically mirrors the key properties of embryonic development in biological organisms. So, like, this is actually a PDF. Yeah, so I just wanted to ask if this is something that would be useful, because like I didn't go through it thoroughly, but like I thought maybe we can if we are monitoring like the development of elegans, and then we can probably use this method as well.
All right. Yeah.
to
Do you
sort
have a
of
better? I mean, I can't open this link. I didn't see. Said my session had timed out.
oh, all right. 1 second. Here's one more link. Can you access this one?
I should be able to. Okay, there. Yeah, a live paper. Yeah, Sebastian Risi. Towards self-assembling artificial neural networks through neural developmental programs. So yeah, this looks interesting. This is yeah. So this is from the A-Life conference last year. So this is where they're doing, and of course, Sebastian Risi's worked on some other things in, like, I think nerfs and other things that are interesting. So the abstract reads here: biological nervous systems are created in fundamentally different ways than current artificial neural networks. Despite its impressive results in a variety of domains, deep learning often requires considerable engineering effort. By contrast, biological nervous systems are growing through a dynamic self-organizing process. In this paper, we take initial steps towards neural networks that grow through a developmental process that mirrors embryonic development. This growth process is guided by another neural network, which we call a neural developmental program, and which operates through local communication alone. Yeah, this looks interesting. I'd like to see maybe if you wanted to propose doing something with this. I don't know if they have their code open source. I guess maybe they do. I would investigate
Yeah, they do
this.
have a repository. I'll share that link as well.
Yeah. So yeah, they're doing using neural growth to sort of build machine learning tools. They're using different benchmarks and optimization methods, and then future research directions and opportunities. Yeah, that would be great. I mean, we've done some of this in our other research group as well. Different approaches to this. Not exactly this approach, because this is something for the neural network community specifically. This is an NDP. This is the repo. So this looks like some details of this paper. So this is their methodology here. This is a neural developmental program. Graph at developmental step T. So this is like a fully connected triangle. There's graph convolution. Then you have graph state S updated via local information aggregation. Then graph growth where you add nodes in. And then edge weights updated. So yeah, they're actually expanding networks. Yeah, this is something that would be very useful, I think, if you wanted to do something in this area. Because it would be useful, I think, for a number of things in the group, not just the graph neural network stuff. But yeah, definitely look into that. And if you would like to propose something like that, that would be acceptable as well.
All right. So yeah, I just wanted to know if I'm headed in the right direction, if this is useful to the project.
Yes, it would definitely be useful.
All right, thanks. There are a couple of more things. Actually, I was documenting all my questions, and then like there was the meeting link, so I was thinking it would be like an hour later. So once I have all the questions, maybe I can text you in Slack. Will that be all right?
Yes, please do.
All right, thanks.
Great. So yes, thank you, Pakhi. Yeah, and so again, we'll be working on Google Summer Code projects. People want to propose things, you can bring them to one of our weekly meetings, or you can propose things in Slack. There are different ways to do that. And I think the proposals are due sort of in April. We're coming up on the time when they're closing, so we have to. I think they're already open, so we need to kind of get proposals locked down. We need to get the outline of the proposal, and then we need to get the timeline of the proposal, which is where you have to figure out what you can do in the amount of time that you have, which is about 12 to 13 weeks. So this is something that is like one of the skills you're trying to build in this program is how to schedule out a project. So, what do you do first? What do you do second? But also, how much work can you do in like a week? And that's always a challenge because sometimes things look easier than they are. Sometimes things are actually easier than they look. So you have to sort of manage your time accordingly.
Yeah, all right. I'll keep those things in mind. Thanks a lot.
You're welcome. Okay. Any other comments or questions? All right. Well,
I don't know.
yeah. Well, thank you for attending today. And we can keep in touch on Slack over the week, and then next week we'll have another meeting. So I look forward to seeing people's progress on numerous fronts here. So thank you. Have a good week.